Hello everyone, this is Monica from second year biotechnology. In this video, I am going to tell about the topic enzymes and classification of enzymes. So, firstly, what is enzymes? Enzymes are simple or compound proteins acts as a specific catalyst, or it is defined as an organic substance capable of catalyzing reaction in the living system. Or enzymes are the biomolecules which contain properties of high molecular mass and specific reactions. Enzymes are biocatalysts. Catalyst means it is defined as a substance that increases the velocity or rate of chemical reaction without itself undergoing any change in the overall process. For example, the student and a teacher relationship may be a good example for understanding how the catalyst works. The student often find it difficult to learn from a textbook on their own. The teacher explains the subject to the students and increases their understanding capability. It is no wonder that a certain difficult things which the students take daily together to understand and sometimes do not understand at all are easily learned under the guidance of the teacher. Here, the teacher acts as a catalyst in the enhancing the understanding ability of students. A good teacher is always a good catalyst in students' life. So, enzymes may be defined as biocatalysts synthesized by the living cells. They are proteins in nature, colloidal and thermolabile in character and specific in their actions. Next, properties of enzymes. These are also called as biocatalysts as they are specific for biochemical reactions. They increase the catalytic reaction rates in the percentage of 5 power 10 to 10 power 20 times than that of unanalyzed reactions. Enzymes have a tendency to offer the forward and backward reactions in reversible process such as generation of ammonia from urea. So, enzymes are more specific in their nature, will be reacting with a substrate. They are clearly affected by solid changes in pH and temperature. Enzymes are mostly active in aqueous solutions. In 1878, Kuhne used the word enzyme to indicate the catalyst taking place in the biological system. Isolation of enzyme system from cell-free extract of yeast was achieved in 1883 by Buncher. He named the active principle as ENZYMES, found to contain a mixture of enzymes which could convert sugar to alcohol. James Sumner first achieved the isolation and crystallization of the enzymes urease from jack bean and identified as it is a protein. Coming to nomenclature. In the early days, the enzymes were given name by their discoverers in an arbitrary manner. For example, the names pepsin, trypsin and other convey no information but about the function of the enzymes or the nature of the substrate on which they act. These are called as trivial names. Sometimes these are known as trivial names of the enzymes, which however fail to give complete information of enzyme reactions. In International Union of Biochemistry initiated the standards of enzyme nomenclature, which recommended that enzyme names indicate both the substrate on which the enzyme acts and the type of action of the enzyme. So, the International Union of Biochemistry appointed the Enzyme Commission in 1961. This Enzyme Commission made a thorough study of the existence of enzymes and devised some basic principles for the classification and nomenclature of enzymes. Since 1964, the International Union of Biochemistry system of enzyme classification has been in force. Enzymes are sometimes considered under two broad categories. First one is intracellular enzymes. They are functional within cells where they are synthesized, and second one is extracellular enzymes. These enzymes are active outside the cell. All the digestive enzymes belongs to these groups. And sometimes the suffix "ase" was added to the substrate for the naming of the enzymes. Example: lipase acts on lipids, nuclease on nucleic acids, lactase on lactose. Coming to classification of enzymes. Enzymes are mainly classified into 6 different types. They are oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. So firstly, oxidoreductases. The enzymes which catalyze oxidoreduction reactions and are classified under these groups. Oxidases, example oxalate oxidase, reductases, example nitrate reductase. Oxidoreductases means enzymes involved in the oxidation and reduction reactions. And second one, transferases. These enzymes catalyzes those reactions which in values transfer of molecule groups from a substrate to reactant. Transferases. The enzymes that catalyze the transfer of functional groups. Means the transfer of functional groups. This is called the transferases. Example, glucose plus ATP gives rise to glucose 6-phosphate. And third one is hydrolases. The enzymes catalyzes the addition of water molecules which later results in breaking of bonds in the substrate molecules. Hydrolysis means enzymes that bring about hydrolysis of various compounds. Examples, maltase plus water gives rise to glucose. And next one is lyases. This enzyme catalyzes the removal of groups from the substrate without the addition of water. Lyases means the enzyme specialized in the addition or removal of water or ammonia or carbon dioxide etc. is called the lyases. Example, butane dicarboxylate plus L-malate gives rise to fumarate. Isomerases. The isomerases enzymes catalyze structural changes within a molecule. Isomerases means it is defined as enzymes involved in all the isomerization reactions. Enzymes are involved in all the isomerization reactions is called the isomerases. Example, glucose 6-phosphate plus gives rise fructose 6-phosphate. Glucose 6-phosphate is converted into the fructose 6-phosphate. Ligases. These are used in their reactions where the molecules has been broken down by the action of high phosphate. Ligases. Enzymes catalyze the synthetic reactions where two molecules are joined together and ATP is used. This is called as ligases. Example, pyruvate plus bicarbonate plus ATP gives rise to carboxylase. So, in this way, the classification of enzymes takes place. The classification of enzymes are 6 types. They are oxidoreductases, transferases, hydrolases, lyases, isomerases and the ligases. So, thank you.